All right, so our next slide is about metabolism. And the first way we should start thinking about metabolism is all living things need to consume. And so what happens to those things that are consumed? What happens inside of the body? That's what metabolism is really about. So down here you can see macronutrients. Macronutrients was an important word, and I told you you all in some indirect way know what macronutrients are. They're the foods that you eat. You have carbs coming from plant products and roots like potatoes. You also have proteins which come from animal muscle cells. All right, the cells or the parts of the body that they use to move. And so there's a lot of protein in these. And so you'll also notice on this Venn diagram that some plants they actually are loaded with protein. And so there are existing proteins in plants that people can get their macronutrients from. And vice versa, now we look on, or not vice versa, but I guess on the other end of this Venn diagram, you see our proteins. Again, so there are some foods that fall within two categories. And you kind of know that already because, right, you know that cheese has a lot of fat. You always hear that eggs have some good fats, we say. Avocado, oil. And so anything that's going to be oil that you've heard of in your life, like olive oil, avocado oil, coconut oil, peanut oil, or just all of these, all the fat liquefies. And so that's what oil is. And so some foods come rich with protein and come rich with fats. All right. And so now my goal for all of you is next week when we start addressing what cells or what makes up a cell, we're going to have to first address what these foods really mean for the cell and where they go in the cell. And so where these carbs, proteins, and fats go, that's our goal for next week, is to find out how every one cell requires macronutrients to build itself. Now remember that your whole entire body is made of cells. And so metabolism is going to be in this class studying how your nutrition is converted into energy and building blocks for the cell, with a special emphasis on building blocks for the cell. And we will learn how we also turn it into energy. So the study of all the chemical reactions, this is the more technical and the appropriate textbook definition of metabolism. It's all the chemical reactions in the body. And so we're going to see next week how they serve the cell. And the last and simple thing I want to move on with is that from now on, or when we start getting into these macronutrients, when they break down, they release energy. And that's going to be important for us moving forward. And so, with that, let's move on. Our next characteristic of life is homeostasis. Homeostasis is a balancing act in your body. All right, so it maintains stable internal conditions no matter what's going on outside. And we're going to use temperature change in your body for our first example. And that's why you can see we listed that one first. And just keep in mind that your body can also do this for things like acidity. So when you think of any person, inside of them, you know that there's a way to cool your body off, right? For example, if the sun's really hot, it raises your temperature, right? It becomes too hot. Here you can see that there are receptors on your skin that will alert the brain, certain areas of your brain that you don't control, like the hypothalamus. And then it's going to cause blood vessels to open wider. That's what dilate means. And so that makes that whole area starts releasing water more quicker. And so that water will be used for sweat. And then so that sweat will then land on your body and cool you off, and your body will return back to a standard about 98.6 Fahrenheit or 37 degrees Celsius. Okay, this is usually the normal temperature around the average. And so that's why if we look at this next here, they match because 37 degrees is what typically on average a human body temperature is sitting at. or 98.6 Fahrenheit. And when we go now this other route, you can see that there's going to be a complete mechanism to respond when it's very cold. So the body is never ever actually perfectly at 37. There, everyone is going to be about 36 point maybe 8. Oh, sorry, 8. Right? Some people might be 36.6 or 36.9, maybe very close, right? 9.1, you want to add more decimals. And so they're sitting very, very, very close to 37. But the thing is that your body, even though it likes to sit at 37, it's never actually at 37. So when it gets hot, 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 that means the temperature goes up. And so you sweat to cool you down, to bring your temperature down. And so if you look at this side, this part will cool you down, and this one will heat you up. All right, so you have two mechanisms in your body, one that is moving the direction of the temperature to cool down, and one that wants it to warm up. This one is about when your skin receptors sense it's cold, they've sensed that it's very cold outside, they then will cause the blood vessels to constrict instead, to squeeze together. And this actually causes muscles to contract. And so it actually also induces shivering, which means you shake when it's cold. And see, that will return your temperature back up when the temperature is very, very low because it's getting very cold. And so this is a balancing act. And so that's what homeostasis is about. Is like, how does this balance maintain itself? And again, it's happening also for pH and other factors. Now, growth and development, not much to say just quite yet in this early on. But when you think about growth and development as a characteristic of life, think about a young child growing into a little girl, growing into a teenager, a grown woman, a more mature, a mother maybe. I'm assuming it's supposed to kind of represent that. And then someone aging and going into old age. And so you see that is a form of growth and development because what she was going to look like at this stage was decided when she was born. And so she grew and developed, right? We go through puberty and developed all the functional aspects of reaching that maturity. And also, when we think about information transfer as well, you can think about plants and, here, this elephant here, we're seeing our elephant produces other elephants. So you're seeing our little tiny one grows into a larger one. That's an easy example of growth. And with that, when we look at plants now, we can analyze plants as well by the way they grow. And also, technically, they also can develop unique features as they grow. Now, viruses. So viruses are going to be a little bit of a topic of discussion for us in this class. And viruses, they exhibit a lot of these characteristics. And so we're trying to make sure we know whether they are alive or not. Are viruses alive or not? With that in mind, if we look at some virus facts, you can see that they are ordered. They have structures, proteins that make up these unique little legs. They store their DNA inside of this capsule, and they have a way of injecting it into a target cell here. All right, so they are very sophisticated, mechanistically, and they can evolve over time. They get over our immune systems. Everyone thinks that it's a scam, that cold viruses and get a cold vaccine. But the truth of the matter is that the cold viruses actually evolves too quickly. So they go through the whole population and come back to you a different virus. All right, they mutate with they just mutate throughout the population. All right, so they do evolve over time and change over time. They can infect a wide range of organisms. And so this means, though, that they cannot produce on their own. And so, I'm reading it incorrectly. And so they cannot reproduce on their own. And so let me give you a little fact about that. When viruses find cells, they actually inject their DNA content, and this isn't perfectly correct, all right? We're going to say their genetic content. It gets injected into the cell, and then it steals all the machinery in here, and then it creates and builds these parts. It'll build that, build this, and then build this. I'm not my best virus, okay? And then it will be able to form itself and then form itself inside of the cell. And it can do, and from one virus can go into a billion virus super quick. And so, little interesting fun fact. And so the question we start with is, would you consider a virus alive? And it doesn't have all the properties of characteristics of life. And so we actually, one reason being is that they do not reproduce. All right, they do not reproduce on their own. They actually just steal machinery to build themselves. And so we say that they exist between living organisms and non-living chemicals, because this is the best way to put it. Non-living chemicals. They have some sort of order like molecules do, but they're just molecules that have gone astray, have gone wild just because of the way they built. Like, yes, I'll just stop there. Next. So when we go through the scientific method, now this is probably the most boring thing to start with in any science class, but when you really go through it slow, you kind of see the worth and the benefit and how you can actually use it to improve your life. So if we look in this direction, this arrow represents an idea, something that you saw, something that you either wish it was true or you want to know if it's true, right? And that's it. And so this is faith growing in your question, that you might have a question that a lot of people want to know the answer to. And so you can prove it and you can trust what you're seeing. So this helps you trust what you are observing in nature. So the scientific method is just basic, kind of like, I don't want to say common sense, but I want to say it's about making sure that you don't believe things without support. That's really all it's about. And so now we observe and generalize. What this means is that we're going to look around, we then get a question. We say, wow, squirrels are active in the morning. That can be a question that you see or saw. And then, you start thinking to yourself, well, I'm not already thinking about what I'm going to calculate to see if it's true. Nothing. It's just like, okay, they come out in the morning. Why would they be only coming out in the morning? And then I see maybe there's nuts on the floor early in the morning. And then I say to myself, oh, maybe they come down to get nuts early in the morning. And that's why they're up in the morning. See, that's formulating a hypothesis already. And you're formulating your idea and the underlying thinking behind the idea. Okay, that's what hypo means. Hypo in Latin means below. So this is like saying the below the question. Why are you asking this? And what is your proposed answer? And so now deductive reasoning compared to inductive reasoning. We'll get more into this in our next week's slide. But inductive reasoning is going to be about not really trying to figure out parts, not trying to calculate things. It's about organizing the idea. And deductive reasoning is about finding the little parts. Okay, finding a bunch of parts to see if you build all the parts, it actually makes the big idea. All right, that's the difference between the two. And if you wanted to see it another way, deductive reasoning is like it wants to know all the parts first to build the whole picture. Okay, and then inductive reasoning, it actually wants to work backwards. It wants to know the big picture. And then as you make your way through this wheel, it's like, okay, what is making it like this? Is it this part, this part, this part? All right, so you kind of work backwards. So with that, you can see now you're going to have to make a testable prediction. That means that you have to test what you're saying. So say what you're saying. So let's say with this squirrels and nuts example, I just say, okay, I'm going to count how many score. One day I'm gonna pick up all the. Let's see, what's a good testable prediction for this? Oh yeah, simple. I'm gonna wake up at this time of day, and then I'm going to count the nuts on the floor, and then I'm going to then count the amount of squirrels and the time of day. Okay, maybe even the temperature. Why not? And then I will see an experiment, observe and see if it's true. I measure it. That's what our experiment is. It's about actually doing the process of getting that information. And then once you have that information, you have to use your inductive reasoning again. You have to see how this applies to a big picture. And does it apply to the original idea you had? Does it support it? And so if it doesn't support it or it does support it, you need to modify your hypothesis accordingly. All right. And you see, you just kind of keep testing your way around that you have enough tests that you can actually have some faith in what you're seeing. Okay. Because you actually have data. And then in this direction, finally, the more you go this way, the more tests you do to prove it, and other people can copy this and see and observe the same thing, then the more you found a truth, which I know can sound pretty dry and boring. But again, I always tell you guys in a biology class, you gotta have to step away and not be like, a general person. You just gotta pretend like someone just threw you here and you don't know what's going on. You're seeing everything for the first time. And how can you trust anything that's created if it isn't at least tested, right? We believe so many things ourselves that we don't really even know they're true. We just say, oh, yeah, I do study better at night because of blah, 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 blah. I am a night owl, blah, blah, blah. But have you ever tried studying really early? Students fall into that trap all the time. Now, with this in mind, if we see each one, I know that going through that without any text can be annoying. So let's just go through the steps to finish this up. And so we make an observation that you notice something in the outside world. You want to learn about it. You see a pattern. You see something that you've never seen before, or something odd, something that's an out of the normal anomaly. And so this is just, what do you want to know? Asking the question is the next step. And so you have to make sure that you're asking the correct question, and then you build a hypothesis. And a hypothesis, again, think of the Latin word, below the idea. It's like saying, what is your idea, and what do you feel is underneath that idea? Right? So here, that's why we start with, this is actually a much more logical step than you'd think. It's not very subjective. For example, here you need to make sure that you're using if and then logic. That means that if I do this, if I remove the nuts from the ground earlier before the squirrels wake up, then I will see less nuts. I know that doesn't make perfect sense, but it's like you're already deciding, okay, if I do this, or if this happens, then this happens. That's really the spirit of a hypothesis, is that what are you trying to connect? And what are you going to try to prove? Now, make a prediction based on the hypothesis. This is that if and then, like I'm talking about, like you need to give like if this happens, then this happens. And then so you can see if X is true, then Y should happen under these conditions, right? This sounds a little bit weird from what we've been talking about, but what this means is like, what if there's a fox? What if there's a fox where those squirrels are eating? That's going to throw off your experiment. So everything should be the same. And so you want to make sure that you even list under what conditions. Okay, so you're really trying to figure if something is true. Figure out if something is true. Now you do your experiment. This is where you're going to be measuring things. You're not really going to have an opinion here. This is the most holiest part of information, okay? Because the experiment is what you observe, the time that it took to measure stuff. Like measuring things takes a long time. Imagine you're doing this experiment for a month. It's valuable. And so this part, you should just be reporting, and that's what everybody wants. Everyone will think you're reliable if you're doing experiments and you're just recording, and you're not being biased. You are just recording. And then, in one scientist's hand, some data could be amazing. And in the hands of another scientist, some experimental data, this data can honestly be useless. Like, like data is important depending on what the scientist is looking for, and can lead to revolutionary stuff. And it happens a lot more than you think. It really, that's why humans are growing so fast, from like, our technology, the knowledge, and how we share, how we learn. It's growing fast only because of our ability to process data. Now, the analyze results, this is the part where, again, the holy part of it, when you just report what you saw, you graph it so that people don't have to be reading freaking sentences with numbers on it. Instead, you just can see a simple graph. When did it start? How much did it grow? And then I do like doing that a little bit in this class. So we will see some graphs, and we will review these the scientific method on that graph. Now, with that, then you have to decide, is your hypothesis supported or not supported? That means, did you find evidence? Do you think it's true? Do you think that other scientists repeating it would be able to see the same thing? But if the answer is no to any of those, then it's not supported. You still report it so that other scientists don't waste their time, doing the same thing. You report it, and then that's useful too. What not to do is pretty useful. It's pretty useful information. And so the hypothesis, however, you don't have to give up. You see something, you see a pattern, you don't know why it's there. And so the hypothesis is about returning to your idea, and then going through the experimental process again. And so you see, good science, I'll say, good science does not ever say believe in 100% or absolutes. Everything can always be improved on, be more accurate. And so since we know this, we just take all, we have to really believe that a lot of people will see the same thing for us to accept it. And so that's what an iterative loop is. It means to try again. It's not anything special. It means literally go revisit your hypothesis. And what else? What else did you miss that didn't support your initial if and then statement? And so I do love this because you kind of see the process, in a different way. And so make sure that you know the scientific method in this order, all right, for this information. That's really what I want you to do. I want you to be able to understand these 5 scientific method principles. And then the inductive reasoning, don't worry about it quite just yet. We'll be playing around with this type of terminology later in the semester, but let's not get overwhelmed. Now, some things that I want you to be aware of going into next week is, when you think about experiments, there'll always be things that you change. Like, okay, you have two plants, and you have seeds in it, and you're letting it grow, and you're letting them grow, but then you want to see if, do plants really grow faster if they get more sunlight? So then this one, you just you have it grow at, say, 1 hour of sunlight a day, and this one gets 3 hours of sunlight. See, you are now creating what we call a dependent variable. That simply means, what are you changing? What are you testing for? And so you can see that 1 hour of sunlight will show me that after a week, if this one only grows this much, that if compared to this one, that after a week it's like this tall, then obviously what I changed was important. And so what you change in an experiment, or what we can also say is manipulated variable, the thing that we change, manipulate, all right, like temperature, drug dosage, anything in your experiment that you decide and you change, independent variable. The dependent variable is the result. That is it, right? Like we saw the growth, right, of my plant here for 1 hour plant. And then on this plant, we have our 3 hour plant, and it's this one. So we got 3 hours of sunlight every day for a week, and this one only got 1 hour. And so you measure, and then so this is your dependent variable. It's what the result was. And in any good experiment, some concepts that are important is that we don't have to rely too much on the text here, but we're looking at what an experimental group and a control group is. And so when looking at this photo, you get a bunch of volunteers. You found what you think is a drug that can reduce blood pressure. And so what you do is you divide your subjects into two equal parties. You don't really know who goes where, men, women, you just kind of mix them up. And then you give one group your drug X and one the placebo. And so the one that gets drug X is considered your experimental group, because you will know that the results are based off of probably that drug. And then the placebo, if you give them nothing, then you know that what you're seeing in the drug X example is significant. Because if you're actually seeing that these individuals have less blood pressure than the individuals here, their blood pressure stays high or it's still too high, then you know that your drug did something. But if your drug, you give it to them the same in the placebo, which can be just like a sugar pill, it's not anything real, it's a fake pill. And so you give drug X, and let's just say their blood pressure stays elevated. Then you give the placebo to the group, and their blood pressure stays elevated. So it turns out that perhaps your drug is not doing anything. So you need this as something to compare it to. And so that's where our control group comes into place. They are going to receive a placebo, which means a nothing, a fake drug. This is so that you can compare the results to each other. And this is an example of how you would support your hypothesis. You create a test, you create a control group, and then you show others that, see, it is my variable. It is my drug. It is the temperature that I'm choosing. That's the reason we're seeing these results. But then the placebo will tell you honestly, will say, nope, I have the same results as you, as drug X. So it's not working. With that in mind, we move on. Now, what information now? You create information and textbooks. You got your textbook. You've probably been scoping through it already. And so how does that information get there? That's what this next segment is about. How does information keep adding onto your population, your group, your nursing field, the fields that you go into? How does it keep being added onto textbooks? That's really what this is trying to get you to understand. So first, when you do experiments, you publish them on journals. You can apply online for scientific journals, and you publish it, and you hope people are working on it. You can blow it up as much as you want on social medias. You can go to labs and tell them that, hey, we're working on the same thing. Look at this information. Look at this data I found. Nobody's doing this yet. And so that's what peer-reviewed journals are. It's people who are working on the same ideas, and you have to be kind of approved, like, overall, but it's not like official. There's not like a judge that says, okay, this is nonsense, this is a good paper. No, they'll say immediately, okay, well, this is a journal, this is some research that somebody came. Go ahead, if it helps your research, why don't you guys test it out? And if it's true, then it will help your research. And so that's how real good knowledge keeps being added. And it's not always fun breakthroughs like, curing cancer, curing some disease like right away. Sometimes it's like, oh, they just found an extra molecule that makes this molecule work a little faster, and we might be able to target that with drugs. It sometimes is not the most exciting thing in the world. But that's what peer-reviewed journals is. It's journals or online publications where people publish and people trust that scientists have looked at it, tested it, and have asked other important people in the field, in that same research field, is it good? They've asked around for people who know what they're talking about, is it worth value? So this is an important word for us. Now, with that in mind, a hypothesis is always just an idea, and it will become a theory if it's been tested a lot. All right, and it also can't always be very specific. All right, it's usually a bigger idea than like the meaning of life is 47. That doesn't make sense, and you're never gonna get ideas. Just don't have values like that. And so that's kind of what broad kind of means like the whole encompassing idea. And it takes into consideration other ideas. And also, it's been tested over and over again, and no matter what you do, it's always right. And this is probably the heart and butter of why all of this is important in your lives and in the life of scientists, because you got to be careful with information. People publish things because they're desperate, they're broke, or something, and they just think that that'll help, or, they're desperate. And they're like artists for ideas, honestly. That's what scientists are. And so, anywho, and so they will be supported over time, meaning that no matter what new information comes, it still holds true. And, it also unifies ideas, just unifies ideas, like cell theory, we'll learn a little bit later about. And so this makes it highly reliable, and this makes it so that when you don't waste your time on bad information. And so some ideas that are controversial, evolution is a big one, but evolution is a theory that hasn't been disproven, has a broad range of facts. It applies to many different fields, different animals, in different domains, right? We learned about domains and kingdoms. And so they are all a result of evolution. And so cell theory now, this cell theory is a little bit about that. We'll get into that a little bit later, but I just kind of want you to keep these two concepts in mind. And remember, what makes a theory? Be able to recognize that on a multiple choice. And so the issue now that you all face is that you can Google something and you don't know what you're reading is good. And now with AI, I guess this part of my information kind of changes because AI now uses peer-reviewed journals. They actually are fed data from scientists and researchers so that it has access to that information. And so I'm going to say, honestly, this kind of is like, I'm kind of spitballing here because things have changed. But before, you Google something and you look for it online and you just, is this good? Is this good? And then AI came and you kind of just trust it. And it's honestly better that you do because they are equipped with peer-reviewed journals. But you can always check the sources, even through AI, you can check where it's coming from. And so what you're always looking for is that it's coming from academic or professional schools and academics, so in the universities, research labs, and then authors. Like, how long has a scientist, there's some people obsessed with ideas in this world and they've done nothing but work on the same thing for like 30 years. And so usually, like, you ask who are the know-hows, right? Like, who knows this field better than me? And what is the purpose of the website? You guys see that right now, we're all the victims of propaganda over and over again, and probably forever. And so everything that you see online sometimes is trying to persuade us. So that's why this upper portion is really important, who you're learning from. And I know that it's hard to believe that some guy, that learned on YouTube can outsmart scientists, but I'll tell you right now, a lot of the stuff you see on the internet is not very good information. Make sure that you're getting information from textbooks and research papers from journal articles. And then lastly, how out of date it is. Like, something from the 1990s is not as good as something from the 2020s. So newer equipment, things like that. So can you trust the information if there's only a lot of old information? And again, where the information is coming from. And so usually you always talk about any information you learn from anybody else, you always give it credit. So that it's like, like being able to, have you guys heard of Bitcoin? You know how they track transactions? That's kind of what it is. It's like tracking ideas. So references allow us to just say, like, when did this come from? Where did this come from? Where did this idea come from? And then if you want to hear more about the idea and why we believe it, the papers are on the journal article sites. And so my hope for all of you is that you grow into critical thinkers. And a lot of times that means just being a skeptic, just first before believing anything. Like, want to test it. Always debate with it a little bit, kindly, politely, to learn, because that'll be useful. Learn how to read graphs. This is my other one. Sounds boring, but it's pretty amazing that there's something that you can look at and it gives you information and the shape of it. You can remember big ideas if you organize it as graphs. And so it doesn't, I personally find it exciting only because I love people becoming smarter. And people that are smart usually can think of just graphs in their heads. And I'll show you that it's not that hard if you organize your ideas when you look at it. And I want you to appreciate the value of statistics. Being able to rely on the information you're seeing is something else. Okay, we'll talk about that later. And distinguishing anecdotes from scientific evidence. This means personal stories. That's what this anecdote means. Personal stories. Let's add that in there. You cannot believe personal stories. You can't even believe your personal stories unless you put it under the scientific method, because then you'll know if it's true. You'll do experiments to see if it's true, and otherwise you might be lying and you're not aware of it. But that is the general existence of human beings, is that they believe what they feel, and sometimes it's hard to swallow the truth. Now, we separate facts from conclusions. And so this is important because, you see something. I'll give you an example. There's a scientist that can make jelly, like found a jellyfish protein that glows like a jellyfish. Yeah, like the jellyfish. And so he took out the protein and he found out what it looked like. And it turns out that it makes the jellyfish glow green. And so that was cool. So once he found that protein structure, that was a fact for everybody. This is the structure of that protein. Green fluorescent protein. And then what happened was that there was another scientist from across the world that said, hey, I could use this protein and I can add it to important molecules and structures, and then we can make it glow, or we can see if it glows, because if it glows, that means that we're able to put markers on other structures so we can track things in the body. So they used it for a different purpose, and all because he only needed the facts. He didn't care about jellyfish. He just saw something that was useful, and he was able to make his own conclusions. Okay, so we'll be working on this indirectly throughout the semester. And understand the differences between correlation and causation. And so correlation is when you're just making comparisons and you're actually seeing similarities. And causation is when you are seeing something, one thing happens, A happens, and then it causes B to fall and hit C. Okay, causation versus correlation. Because let's say that because this always happens, so what if every time you do A, you see C and you never saw B? So you're always going to think every time I do A, I do C, or C happens. But in fact, if you don't have all the causation, all the experiments to prove it, then how do you know from A to B, right? Now, pardon, this causation teaches you, okay, this actually causes this, and this is what causes this. But now I get that every time if I did this, it looked like I did this. All right, that's an example of a mind mapping, that kind of situation. All right, with that, the last things to add is that all those research papers that you see, they're not made up by some crazy guy in his lab. They're normally not accepted into journals unless they have appropriate sample size, meaning they have enough people to really say, okay, yes, this drug works. But if you only have one person in your sample, how useful is that? And so control group, you always need to know a group where you gave the placebo, or that they're not supposed to have anything happen. Nothing out of the ordinary. of the ordinary. All right, so that is what a control group is all about, is making sure that you know that what's supposed to happen. Now do your thing, and then let's see what happens. Next, most of the time, in most journals do not accept research if it hasn't been tested by other labs, that they have to be able to also repeat the same results, so that way, you can't have people pulling a fast one. And trust me, I've been a victim of that when I was trying to do research in my my research days. And man, I tell you that I spent months trying to do an experiment that these guys from Harvard were claiming, and it didn't work, and it turned out to be nonsense. So I wasted so much time and money and resources on that. And so this is a real one. All those people are like, oh, I discovered this. I bet you they can't have like 3 or 4 labs prove it. So you always got to be skeptical. And the last thing to add is all these journals that I'm talking about, science and data, these will usually be on scholarly articles, right? You normally have to go to Google and you write in Google Scholar in the search bar and it'll take you there. And so the scholarly articles, usually it'll tell you from where they come from, the university they come from. It's always somewhere in that writing. And primary sources are pretty much researchers and scientists. Secondary is when they combine their works. Now that they have a paper from Switzerland, a paper from this part of the world, that idea, once everyone's proven it, they then put it into a textbook. And that's what a secondary source is. It's a text, like a textbook, a review article. And so that is what secondary sources are. And lastly, publicly available Wikipedia, which does okay, right? It has some sources, but can you trust it? That is the tough part. My friend once, back in the day, before they made Wikipedia with more regulation and you were just allowed to upload whatever you wanted, my friend was the president of a made-up country on Wikipedia. So, you always got to be careful with what you read on there. And so peer review overall is the main highlight or the word of the day here, that you want to make sure that your work is always evaluated by people who are impartial. That means people who are not biased. Not biased. It means that they're willing to listen to new ideas and they're not trying to be right. They're really open to things. And then qualified, they obviously should probably at least been in the field for a while. It's not like a movie. There's so much going on that honestly, you just can't listen to everybody. And lastly, you don't know who's going to evaluate you. You don't know who made the paper. So if an enemy school with another school, their competitors, they do amazing research and then they'll send it to Harvard, and then Harvard will look at it and they don't know where it came from. They just have to honestly review it. So, the information online, you can trust peer-reviewed papers that at least people are trying, okay? And there has been abuses. I know there has been abuses, and I know you've heard of some of, but that is the reality of life, is that what information can you trust? That's very valuable, and more valuable than money and treasure. All right, with that, thank you very much. Make sure to read the rest of the announcement for the discussion portion of today's lecture. Take care, everybody. I'll see you on Tuesday.